Thank you. Brooke is a hard act to follow, but I'll try. She did the work she reported on in my lab, and I'm so proud of her. She did a great job. I'm going to talk about the rest of the project, but I'll use her introduction as a starting point for mine. Essentially, she did the control studies in a larger project that involved giving intranasal pDNF nanoparticles to the rat by the intranasal route of administration, and our goal was to see whether we could improve that delivery and transgene expression from that plasmid. So these were the project goals. It's similar to the slide she showed. We're basically looking to see whether focused ultrasound can improve the delivery of these nanoparticles and the amount of GDNF that we make in the brain. She showed a similar slide about our approach. Essentially, we gave 2 rounds of sonication on either side of intranasal delivery of, in our case, the gene nanoparticles. One thing to note in this case is that the GDNF plasmid has linked to it an EGFP reporter so that we can detect in sliced sections or in ELISA assays the presence of the transgene product that was expressed from this plasmid rather than just GDNF that's endogenously present. And as you can see, in the same way that Brooke just described, there are 2 sonication sites, one on the right in the B section and second on the right in the C section of the rat brain. Okay, we demonstrated that there was blood-brain barrier disruption using T1 weighted imaging, and there was no difference between the 2 sites in the degree of that contrast enhancement. So essentially both sites received the same treatment. Okay, this is a slide that again shows the scheme of our study, and the first box I'll talk about is whether or not focused ultrasound increased transgene expression, levels of the protein measured by ELISA. And as you can see in this slide, that on the left side, all of the right regions, the A right region, B right, C right, are significantly increased in their expression of EGFP, which is the marker for GDNF, and on the last section, the D section, there was not a significant increase, but that was not a sonicated area. If you group all the data in terms of the right and the left hemisphere, the right hemisphere had significantly more EGFP expression than the left. So the next point was to see if we could see what cell types were transfected in these brains that had received the nanoparticles intranasally as well as focused ultrasound. And there's a little primer here on how to understand the images that follow. The transfected cells, the ones that had taken in our plasmid and produced EGFP, are always going to be stained red in the subsequent slides. There's some red cells on this image. A second cellular marker for different cell types that we were labeling for is always going to be shown in green. So in this case, we have rat endothelial cell antigen, which is labeling endothelial cells. Always stained green in this case, and you can see that there are red cells, transfected cells, that are lying right outside the vascular endothelium. And they have the morphology that made us believe at first that they are pericytes, P-E-R-I-C-Y-T-E-S. And this would be what those cells would normally look like. Okay, in the next slide, you can see what we found when we looked for transfected cells, the red cells in the brain, and looked to see whether they co-labeled with markers for RICA1, GFAP, which is an astrocytic marker, and NUN, which is a neuron marker. And this is in normal animals that did not receive ultrasound. Oops, not to go forward here. They were not RICA positive, the red cells were not GFAP positive, and they were not NUN positive, so they were none of those cell types. The next thing we did was to look to see what ultrasound did to change this pattern, if anything. And again, this was in animals that did not receive ultrasound. In the next slide, you can see that when we did deliver ultrasound, there was a change in the pattern of red cell expression. These cells were in clumps, and they did not appear like this. Red cells were never in clumps like this, except on the side where we had delivered the ultrasound. Again, they were not RICA1 positive, they were not GFAP positive, and they were not neurons. And so they are some other cell type. And it was at this point that Brooke came along and decided to do the assay for IBA1 to see whether the cells might be microglia. And we discovered that in fact they are. Here's an image that is stained for IBA1. The red cells exactly overlap with the green, and it looks as though some, at least, of the microglia that are recruited to the site are also being transfected by our plasmid. So, when we measured the percentage of those cells that are transfected, we found that greater than 70%, somewhere between 70 and 75% of the transfected cells are microglia at sonication sites. And that is a far smaller proportion or percentage if you look at non-sonicated regions on either the left or the right side. So it appears that we are transfecting microglia as the predominant cell type, at least at sonication sites in the brain. So our conclusions are that we think that focused ultrasound does improve delivery, does increase tissue penetration of the nanoparticles, and it changes the pattern of transfection so that we're transfecting microglia largely at the sonicated sites. We think that the implications of this are still unclear, whether or not microglia serving as the source of an endogenous neurotrophic factor is a good or a bad thing, but that is the nature of what it was. And we're optimistic that this kind of focused ultrasound treatment might improve the delivery of these nanoparticles to the target areas for Parkinson's disease. Thank you.
This is some fascinating work. For those of us simple clinicians, can you just put together in perspective the biology of why an intranasal delivery is preferentially going to pericytes without FUS, and with FUS it's going to microglia? Like, what's the mechanism underlying this?
It never involves the blood-brain barrier. The intranasal delivery bypasses the blood-brain barrier, and it allows the nanoparticles to enter the brain and enter cerebrospinal fluid, which circulates in the brain. And the nanoparticles then move through basically perivascular channels, and they reach sites that are close to the blood vessels on the outside of the vessel. And those are the first cells that encounter the nanoparticles and the type that get transfected. Usually the cells that we see transfected are next to blood vessels. It's only after focused ultrasound that there's a deeper penetration and microglia seem to be getting transfected. Normally it's these perivascular cells. They could be parasites, they could be vascular microglia, or vascular macrophages that would label for IBA1.
First here and over there.
Yes.
Can you tell us a little bit more about the composition of the particles? Is this a PEI non-viral particle you're using?
Yes, it's a non-viral particle. It's made by our collaborators at Copernicus Therapeutics. It's a polylysine compacted DNA nanoparticle, so it's not at all a viral vector.
Right. Could that be responsible for some of the microglial activation of the polymer
It's possible.
itself?
I mean, I'm not certain of that.
Thanks, over there.
I have a question. When delivering intranasal, by intranasal delivery, usually part of the dose of the drug goes per os, goes into the stomach.
Yes.
Did you check other organs than just the brain, or?
In other studies, we have shown that when we radio-label GDNF and give it intranasally, that a large amount of it does go into the stomach. We didn't do that with these nanoparticles, but we could assume that's the case. And our collaborators at Copernicus have tested that with different nanoparticles of the same composition, and they found that there's not transfection to any appreciable extent in the lung or in the liver or stomach. So I think we're okay on that.
Yeah, thank you. We are traveling well with time. Thanks again.